Okay guys, so this time we're going to do a monohybrid genetic cross. So if I just put monohybrid, so the prefix mono means one gene. So for example, it might be the gene for the height of a plant. So we could say height of plant. Now because the plants, imagine they were diploid, that means they've got two sets of chromosomes. So two sets of chromosomes. So that means the parent plant has two alleles for this particular gene. So for example, if I said we had a heterozygous plant and it was a tall plant, it would have two alleles. One would be a dominant allele, which means that the plant will have a dominant phenotype for this gene, and the second allele is recessive, which is not expressed in the phenotype. So this would be what we call the genotype, which is the combination of alleles that gives rise to the physical characteristics. Now, if we were to cross two plants and they were both tall and they both happen to be heterozygous, the first parent would have that genotype, and then we put a little X because we're going to cross it with another parent plant that's also heterozygous. So the first thing we have to do for any monohybrid genetic cross is to write down the parental phenotypes. So I'm just going to put parent 1 is a tall plant and parent 2 is also a tall plant. Now the second step of the 5 is to write down the genotypes that we just discussed. So both heterozygous for this particular gene. We've got two alleles, one dominant, one recessive. And the third step will then be to write down the gametes and which allele the gametes will possess. So any parent plant will make 4 daughter cells through meiosis, but there's only going to be a single allele inside each gamete because the gametes are haploid. So if there's only one set of chromosomes, you've got one chromosome with that gene and therefore one allele. The parent cells that underwent meiosis, for example, the microspore mother cells in the pollen sacs, or it could be the megaspore mother cell in the ovary of the carpel, they would contain two alleles. So I'm going to put my gametes in circles. So these gametes on the left are from parent 1 and the gametes on the right are from parent 2. So I'm just going to put gametes. Now the next step will be to do a Punnett square. So we're going to take the 2 possible gametes from parent 1 and we're going to cross them and fertilise them with the 2 possible gametes from the second parent. So step number 4 is going to be a Punnett square. And the way I'm going to draw this is parent 1 on the left, and then I'm going to have Punnett square like this. Probably best to use a ruler. I'm going to put a dominant T and a recessive T, and then I'm going to have parent 2 on the top. And again, parent 2 makes a dominant T gamete and also a recessive allele gamete. So recessive allele here is for short or shorter growth. And then I'm going to get fertilization. So in the Punnett square, we do our theoretical fertilization. So one of the theoretical offspring will be homozygous dominant. The second theoretical offspring will be heterozygous. The third theoretical offspring will be, again, heterozygous. And the fourth theoretical offspring will be homozygous recessive for that gene. So the last and the 5th stage is to write down the offspring phenotype ratios. So if we look at the offspring, this is in theory, isn't it? We've got 1, 2, 3 of the 4 that would have a dominant phenotype, because the homozygous dominant will give a dominant phenotype, and the heterozygous will also give a dominant phenotype because of the single dominant allele. So we're going to say 3 tall plants to every 1 recessive plant. So that would be the short phenotype. So of the 4, 3 of them will be tall and 1 of the 4 will be short. So it's a 3-to-1 ratio of tall to short.